We can resurrect extinct animals and smell the scents of flowers that went extinct thousands of years ago, all using a technology called genetic engineering. We grab the organism's DNA and identify a gene that controls the trait we want. To snip it out, we use a restriction enzyme like a molecular scissor. For example, 𝔼𝕟 cuts DNA at certain points, but it also cuts the DNA at similar sites, leaving us with multiple DNA fragments. Next, we have a circular DNA called plasmid that bacteria love to carry. We cut the plasmid with 𝔼𝕟 and then glue in our gene with DNA ligase. The plasmid also has a special marker that makes the bacteria resistant to tetracycline, an antibiotic. Some plasmids take up our desired gene, some take up other DNA fragments, and some just snap back together. Next, we take E. coli, which die if exposed to tetracycline. To get the plasmids inside, we give bacteria a quick cold shock, followed by heat, creating tiny pores in their membranes. Then the plasmids slip in. Next, we plate the bacteria on tetracycline agar, basically fancy antibiotic jello. Only bacteria with plasmids carrying some kind of DNA will survive. To figure out which ones got our gene, we extract the plasmids and cut them again with restriction enzymes. Different genes are of different sizes, so we run them on something called gel electrophoresis. The gel separates the DNA fragments, showing us which plasmids took up our target gene. With genetic engineering, gray wolf genes were edited to create pups with dire-wolf traits, and scent genes were reconstructed from extinct preserved flowers to recreated scent, like the haunted rose and grassland opera.
We can turn microbes into biofactories to produce medicines, sustainable fuel, to no-meat burgers. We can also genetically modify viruses to selectively attack cancer cells. Today, with genetic engineering, we can reimagine what's possible.